I'd like to talk about time-gated Raman spectroscopy, a cool tool to overcoming fluorescence hurdles of biological samples. So let me introduce our company, because I'm not from Timegated Raman. I'm from analyticon. We are a specialist for on-site analytics. And that means we either work with these portables that you can see on the top two pictures, for example, raw material ID or checking for hygiene monitoring, or we work with sensors that are integrated into the process. So like the near-infrared spectrometer on the left or the Raman spectrometer on the right. And as we talk about time-gated Raman spectroscopy, I want to briefly address Raman spectroscopy. I won't read that because I only have 5 minutes, and I'm quite sure you all know Raman spectroscopy. And I mean, Raman is great, yeah, for a lot of reasons. But there's an elephant in the room, and I'm quite sure that most of you know this elephant, and this elephant in the room is called fluorescence. So that does mean that you don't have this nice spectrum you saw on the slide before with a flat basic and cool peaks that are rising out of this background, but you have something like that, what you see on the slide. So there's an overall background that is covering your Raman signal, and that's really a topic in pharma and chemical industry, and indeed in biopharma. So what's going on there? Why we see that? So on the left, you see a plot with the typical spectrum, and what you can see if you compare to the time that the Raman scattering is coming to another time than the fluorescence. It's not a big difference, but there is a difference. And how would it be if we would be able to set some kind of a time gate on our spectrum and just focus on the early signals? And that's exactly what Timegate is doing. They have the possibility to make a real fast gating on those signals, and then you see that in an improved signal. Yeah. So that's what they do, and it's even better. So I said fluorescence is an issue in pharma and biopharma, but that's not the complete truth. To be honest, fluorescence also transports some information. And the cool thing is that Timegated added a third layer on your data. So it's not a two-dimensional plot that you are familiar with. So with the Raman shift and the signal intensity, they just added a third dimension, and that's time. And you decide whether you want to see only the early signals at the early time, or you go later in the signal and also want to analyze the fluorescence. So you have both. So which settings we can provide? That's a very conventional setting. That means you have an immersion probe that's, for example, used in the lab, or you integrate the probe via a shot viewport or a PG35 into your reactor. Yeah, so an immersion probe. But what's also very cool is that we are able, or Timegate is able, to offer you a setting where you can make spatially and localized measurements with a microscope. So you focus on an area on your sample, and you just get Raman spectrum for that area. Yeah? And the third setting they offer, quite interesting, I think, for some kind of research and development or formulation development, that's plate reader. Yeah, so you have the opportunity to really make high throughput screening, measure the individual particular wells, and get Raman spectrum only from the well you are interested in. So some applications, quite clear. Most interesting for you, I think, is the biopharma upstream processing. On the left, you see conventional Raman, and in the middle, the plot shows the time-gated Raman spectrum, and it's clearly seen that the spectral quality improves, and that means at the end, of course, you improve the way you can monitor your process. Yeah. At the end, it's all about avoiding batch loss if you're working under an industrial regime, and to increase your product yield. But also downstream is quite interesting, because compared to UV-Vis, what you can see on the left, Raman spectra are really also possible to differentiate between different things. So UV is saying there is coming something, and Raman spectroscopy can also tell you what is coming. Yeah, so you can focus on that. So I'm even faster than I expected. I'm talking so much, but then we have more time to discuss.